So today we are going to start differential gene expression linked to the process of development. And as we yesterday learned that after fusion of sperm and the oocyte, the two haploid gametes, the 2N, the diploid zygote is formed. This zygote undergoes many cell divisions, mitotic cell divisions, and by the time it reaches blastocyst stage, it gets divided into two major kind of cell types. And one cell type we said were the trophectoderm or trophoblast cells, and the other cells we yesterday also looked at video is the cluster of cells which is referred to as inner cell mass. All our body comes from the cells which are in this inner cell mass, and yesterday we learned that the onset of gastrulation shows us massive movement of the cells, and we have, as a result of gastrulation, we have ectoderm, mesoderm, and endoderm. So these three layers of cells are called germ layers. And this distinction is because different. Now these cells, ectoderm, will lead to formation of your epidermis and brain, etc. Mesoderm is going to lead to internal organs, and so the endoderm. So this distinction is actually only possible when we have differential onset of differential gene expression. For example, if the total collection of genes in a nucleus is A to Z, these genes are differentially expressed. A may be the developmental genes. 80% of the genes, they are normally considered to be housekeeping genes, and housekeeping genes are the ones which maintain normal homeostasis of the cell. They maintain, ATP synthesis and, the day-to-day affairs, everyday affairs for the survival. That's why they are called housekeeping genes. The rest of the genes which are developmental genes, key developmental genes, they are selectively switched on and switched off. And that selective gene expression is called, differential gene expression, which leads to cell fate determination. Which cells are going to become ectoderm or mesoderm and endoderm? As the word beautifully describes, cell fate determination. The fate of individual cells is getting determined. And once cell fate is determined, the next step is let's say here we have earliest single cell, which is the precursor and is going to make heart. This cell is going to continue divide mitotically. Genes are already heart specific developmental genes are already expressed. They start expressing in the cell, and when the cell mitotically divides, it then leads to maintenance of this differential gene expression after every mitosis. And then we have a couple of 100,000 cells. All of them, they belong to what we call this cell lineage, which is of heart cells. Through mitosis, these genes are maintained in the off state And eventually what we see that these cells differentiate, and here they may be of different cell shape, but here the shape of the cells, it start changing, and these cells, they eventually look like this, and we see process of differentiation in which we see eventually heart is formed. This is terminal differentiation. So today we are going to talk about this very interesting phenomenon. We'll quickly go through this process of development, that during early development, this is from where on the day one we see our development starts. Somehow by this time we already gone through cell fate determination, and differentiation has started. And all the future progenitor cells, they are going to remember their original identity. Now, towards the end of last century, there was a very interesting experiment they did, which resulted in cloning of sheep, Dolly. The whole idea since last 60 years was that, as we just learned, we have single cell, and then this is a zygote, and from this single cell, we have 200 or more than 200 different cell types in our body, and they all attain different identity. The idea was that each cell type contains same 46 chromosomes, but the cell's identity is different. Cells, they develop differently. They contribute to different kinds of, cell types. So what goes from here to here in the genome, has the genome undergone some irreversible changes that, cannot be erased, and cell has permanently committed to certain cell lineage by modifying the genome in such a way that these 46 chromosomes, or the genes lying on these 46 chromosomes, they cannot go back in their life. So what they did, they simply took skin cell, the diploid cell, this is the skin cell. They took the udder cell, which is a somatic cell. They took udder cell, the somatic diploid cell. They took its nucleus, and after removing this nucleus, they transplanted this nucleus in oocyte. So they removed the nucleus of the oocyte, okay, replaced the nucleus of the oocyte with this diploid nucleus of a somatic cell, and then they implanted this in the uterus of a foster mother, and this tried to see if this diploid nucleus, which now does not involve fusion of sperm and egg, it leads to development of a completely new baby. This gave them conclusion. This concluded that the genome, the somatic cell nucleus, actually it has not undergone some irreversible changes. All the chromosomes and the genes lying on the chromosomes, they are, switchable. We can reprogram them if we provide them in the right environment, which is a cytoplasm of oocyte. So it means the cytoplasm of the oocyte or the egg, it has some ingredients which can support development of completely normal animal, a baby, in the presence of diploid nucleus. So it means during the process of this development, this differential gene expression, which is established and maintained, it does not involve any irreversible changes in the genome. Okay? So let's now look at different stages of development, because we have to look at the bigger question. We have to look at the context in which we are going to understand today's lecture. So we start our development here. This is the first day of our life, second day, third day, and this cell is dividing mitotically. 7th day, 8th day, and you can see, you know, this is like embryo is getting changes in some, you know, shape. Up to here, 7, you just look like, you know, maybe a clump of cells. But here, you know, some movements of cells and change in overall embryonic shape you can see. Then by 19 to 25 cell stage, 24 to 25, and here you see, you know, by 23 to 25, and this is the organogenesis stage. This one is what we saw, organogenesis, or which was beginning of neurulation. By this stage, which is 17 to 19, or, you know, 23 days, you already have signs of cell fate determination. Cells, they start committing to different cell fates, and this happens due to differential gene expression patterns. And I told you differential gene expression patterns means you are now having selectively cell type specific gene expression patterns. For example, here already the cluster of cells which are to be made eyes in future, they will start expressing eye specific genes. Heart, the cells which commit to heart, they start specifying and they start expressing heart specific genes. The muscle cells, they will start expressing muscle specific genes, and so on. जी दानिश,
जी सर, मैंने पूछना था कि सर which organ के cells start developing first in the human body, मतलब जब in the
which organ?
So which organ starts developing first, मतलब?
Yeah, good question. So normally morphologically when we see and the earliest sign of, you know, organ, what we see in first few weeks of development is the heart.
जी sir.
You know, because this is the evidence we see when we go to gynecologist and they do the ultrasound, we can see the, you know, the cells. It's not heart, the fully differentiated heart, just few cells, they have started beating.
Sir, so they're specializing into becoming the heart.
Yeah, yeah.
Okay. Okay, sir. Thank you, sir.
So yesterday we already talked about these processes: determination, differentiation, morphogenesis, and growth. So these are, you know, cell fate determination. Then you see, you know, already here differentiation and organogenesis has started taking place. And through these changes in morphogenesis means, you know, creation of an order. You are becoming an orderly structure here. And from here onward, it's just growth. Embryo, after going through differentiation and morphogenesis, it's just growing in size. And the massive size or the massive increase in growth of embryo, it takes place during last 3 months of the embryo. We already covered in great detail yesterday all this, so we are not going to go through again, but This is just recapping what we went through. Now, how cell fate determination or how fate of cells is determined. This is a very, very beautiful phenomenon in biology. This is already, you must have appreciated looking at the example of Dolly, that, you know, how a skin cell, you know, which has undergone massive changes in gene expression, which is fully differentiated, which has become skin after so many months of development, years of development, and it's a terminal differentiated state. Terminal differentiation means these cells are never going to divide again. And you know, you take its nucleus and implant in oocyte cytoplasm, the nucleus forgets everything. It goes back in lineage, it gets de-differentiated, and you know, gives you a totally new, normal, healthy individual. So the cell fate determination is one of the fundamental processes, one of the beautiful, you know, phenomenon in biology. I would say it demonstrates some of the beautiful marvels of engineering in development. And one example is here. For example, we are starting development in Xenopus in frog. We had yesterday, we did talk about, you know, we have thick yolk on the vegetal pole, and on the animal pole you have the nucleus. Now what happens, you have this cell undergoes fertilization, and then after mitosis we have two cells which are a result of symmetrical cell division. When we say symmetrical cell division, because the two daughter cells, they contain same number of chromosomes diploid 2n, and the cytoplasmic contents are also equally distributed. This is simplest definition of symmetrical cell division. Now from here on, when it divides, look, it's now asymmetrical cell division. Two of the cells, they are differently in terms of their composition of cytoplasm than the other two. Already difference is created, asymmetry is generated. Like yesterday we talked about degradation of beta-catenin by GSK in only a small region of zygote after fertilization, and asymmetry is generated. Now this cytoplasm is, you know, is a beautiful structure. The contents in cytoplasm are amazing because, you know, although we talk a lot about chromosomes, genes, you know, the gene expression, switching on, switching off, but we don't know how they are switched on and switched off, from where the communication comes in, and the key holds in actually the cytoplasm. There are so many cell communication molecules here, because when these two cells divided, we had many proteins, many thousands of proteins which are performing their function here in the cytoplasm. And when cytoplasm is asymmetrically distributed, so many proteins are now harbored by these cells, which may be missing in cytoplasm of these cells, and the nucleus contains same set of chromosome and same genes. So the cytoplasmic contents here, they have created an environment which are going to regulate these nuclei different than the cytoplasm of this cell. So this is kind of earliest, cellular processes which play a role in cell fate determination. I explain you on this slide how this asymmetrical cell division or asymmetry may lead to cell fate determination. For example, when we are in early stage of development, we already talked about beta catenin degradation. So here GSK was depleted or degraded and we had beta catenin, and here we had minus beta catenin, no beta catenin in this region. When this cell is going to divide, you have now two cells, one with beta catenin, the other not, and beta catenin is a transcription factor. The cell which has beta catenin, so transcription factor is going to go inside the cell and activate genes. And this one, although it contains all the genes which are activated by beta catenin, but due to absence of beta catenin, these genes, they remain off. Okay, and there are hundreds of genes which are regulated by beta catenin. So this is a common phenomenon in early development in Xenopus, in humans, in flies. We will soon see what happens in flies. So when you have two different cells early on, now you have, and then these cells, they keep dividing. They will look like similar to their mother cell. These two cells look similar to their mother cell, and so on. Now what happens that two different kinds of cell types, so one is, let's say this one, this is other one. Now you see this one is having these receptors on the cell surface, this one and this one. Although they look similar at the cytoplasmic content for a while, but the inducer, a cell which is sending signal, this is better perceived here. Look, this signal is fully bound to this cell as compared to this one. The cell surface receptor is not fully bound, and there could be many reasons why this is happening. Now this cell surface receptor bound to this ligand is now going to send signal into the cell, into the nucleus, and you will have this inducer bound onto a specific gene. Same gene is present in this cell as well in the nucleus, but since the communication is almost very low or nothing, we don't see this gene switched off, rather it remains off in this cell, but the same gene is switched on. Messenger RNA is synthesized, protein is synthesized, and then this is the onset or beginning of cell fate determination. In terms of early cell development, there's a beautiful phenomenon in fruit flies, and the fruit fly embryo, yesterday we looked at, It's a syncytial blastoderm. You may remember that, where a number of nuclei are there sharing the same cytoplasm. All these are nuclei, the dots are nuclei. They are dividing, they are all sharing the same cytoplasm. Now, when you go to, a certain developmental stage, we said these cells, they start becoming cellularized. Nucleus, each nucleus, it starts having its own cell membrane, so they become cellular blastoderm. This stage of syncytial blastoderm, when nuclei are sharing the same cytoplasm, is an amazing phenomenon in development. Why? Because all the nuclei share the same cytoplasm. Whatever are the contents of the cytoplasm, they are available to nuclei equally. However, if we look at fly embryo as a whole embryo, as a cylinder, so the nuclei which are present here along the length, what we learned during early 70s and 80s was that the mothers, when egg is being formed in fruit fly, mothers, they deposit some messenger RNAs here. Each line represents a specific messenger RNA which mother is depositing at the time of egg formation. Also, mothers deposit here as well specific messenger RNAs. And we call these messenger RNAs, or genes for which mothers deposit their messenger RNA at the oogenesis or egg formation in their ovaries. These genes were named maternal genes because mother is depositing. They are present in father as well, but at the time of oogenesis, mother is already like depositing food or nutrition for its baby. Look how caring mothers are. They not only provide you the mitochondria, all your future energy, but your mothers already deposit some messenger RNAs. Now, what is the function of these messenger RNAs? When fertilization takes place, sperm hits the oocyte. Zygote starts, having mitotic cell divisions. There is no transcription of zygotic genes, no transcription. TXN means transcription of nuclei in zygote because cells are just dividing through undergoing cleavage divisions. And what happens, the messenger RNA, as soon as fertilization takes place, the messenger RNAs which are deposited by mother, all of them, they are translated. Their translation starts. Protein synthesis starts. All the messenger RNAs which are anchored here, they are fixed physically with cytoskeleton anchored. Their proteins, they start being formed. And proteins on this side, they start being formed from these messenger RNAs. So this side, the head side is called anterior, the tail side is called posterior. So there are proteins which are present at anterior side and proteins which are present at posterior side from maternal messenger RNAs. Now, since it's a one big cytoplasm, these proteins, they freely start diffusing in the cytoplasm. And since it's a simple diffusion, you can imagine you will find maximum concentration of these proteins here. And the anterior ones, their concentration will become less and less and less and less and less. Like this. Highest concentration is of these proteins, maximum on the anterior side and least on the posterior side. Same is the case for posterior ones. Maximum concentration of these proteins will be here, and you will have like this. Highest concentration, least concentration, and highest concentration and least concentration. Now the nuclei which are spread all across this embryo here from anterior to posterior, they are being exposed to different concentrations of these proteins. And based on the concentration of different proteins, these cells, they start expressing different genes. For example, cells which are exposed to very high concentration of maternal genes on the anterior side, they may look like this. The cells which are here in the middle, they start expressing genes, different genes, because the concentration gradient, if this is a, for example, gradient of a one protein, highest to the lowest, the cells which receive, so based on the concentration of this, it expresses or unable to express certain genes which are present here but not there, but or present here but not there, and so on. And you see what in developmental biology we call French flag model. So these proteins, we call them morphogen gradients. This is a gradient, no? Anterior of embryo, posterior of embryo. Let's say this is gradient, maximum to minimum. And on this side, let's say posterior to anterior, there is another protein, maximum to minimum. So these proteins are called morphogen gradients because they are involved in morphogenesis, in creating an order in the process of development. This is an order, a beautiful order. What is the, I come back to this slide later on. This slide should have been later. These morphogen gradients were characterized and discovered beautifully in fruit fly, and we learned that right after fertilization, you know, these morphogens, they start expressing, which are deposited by mother, and the result is all the nuclei which are present in the syncytial blastoderm, they are exposed differently to the different concentrations of these maternal genes. And as a result of cell fate determinations, you see segmented body plan. Embryo is divided into 14 discrete segments. And these segments or segmented body plan is fully preserved from here all the way to here. These three are fused head segments, The next three, this one, and this one, are this thorax. And then you see these segments, A1, A2 are fused, 1, 2, 3, 4, 5, 6, and then fused. These are these segments, abdominal segments, or 8 abdominal segments. This is not possible, simply not possible, if we don't have these morphogen gradients and the process of differential or cell type specific gene expression established already here. Let's see how it looks like. Here is just for simplicity, there is a maternal messenger RNA shown. Only one is shown, there are many. The name of this gene is called Bicoid. Mother deposits this. As soon as the fertilization happens, the sperm fertilizes. This messenger RNA is translated. And since it's not just one molecule, there are many molecules of messenger RNA. You see a massive concentration of bicoid, which starts diffusing. And what we see, we have on the anterior side, maximum concentration, and on the posterior side, minimum. And all the other anterior or posterior morphogen gradients will be established. Now, what is a bicoid? Bicoid is basically a transcription factor. Transcription factors are proteins which bind DNA and help in switching on those genes. So bicoid binds many genes and then activate those genes. Now, what are those genes, for example? Here we had only one. Now we are going to see two contrasting examples. You see, this is a real picture, a staining of bicoid maternal messenger RNA in the zygote. When you have fertilization, you see this blue line is the bicoid gradient. Protein has diffused. The messenger RNA of bicoid, remember, is just anchored. That's why you see messenger. This is messenger RNA staining. It's fixed, physically anchored. But when protein translation takes place, it diffuses freely. And this red line is basically the messenger RNA signal. Now, on the posterior side of embryo, there is another protein, maternal protein, which is called nanos. You see the nanos, the posterior to anterior, and you see the gradient of nanos. This is also a transcription factor. Now they do opposing function, they do opposite role. What they do, for example, since I told you they are transcription factors, bicoid goes and bind at the DNA region of a gene called hunchback. And switch it on. And then the nanos tries to suppress this so that the hunchback is only present in the anterior 1/3 of the embryo and not towards most posterior silent. You see what is happening? the cytoplasm, because it's just one big cytoplasm and nuclei are there. Cytoplasm is having different morphogen gradients, and nuclei are being exposed to certain threshold gradients of those morphogens, and then those nuclei attain different identity, or they become specialized based on their position in the embryo due to the morphogen gradients they are exposed to. Now, what is happening actually? All this is sign of long-term developmental plan. This is the earlier stage. The genes are called maternal genes. Bicoid, nanos. So for example, bicoid, nanos, caudal, etc. You don't need to memorize those names. Immediately after fertilization, their gradients are established. For example, these are the anterior genes. On this side, I draw, let's say, gradient of posterior genes. Now, since these are transcription factors, they go and bind within the nuclei on DNA on their specific targets, and they activate specific genes, the next class of genes, which are called gap genes. Remember, these maternal genes are transient. They are proteins. They degrade in 30 to 45 minutes. They disappear. But they set up the stage for long-term developmental plan by activating, switching on gap genes, which are themselves are transcription factor. And they are also transient for 30 minutes or so. Gap genes also are, moving freely into the cytoplasm and nuclei according to their specific position in the cytoplasm are exposed to different concentration of gap genes, which means genes in those nuclei will be differentially expressed. Eventually, what you see when gap genes disappear, before they disappear, they activate next cascade of genes, next class of genes, which are called pair rule genes. They are also transcription factors. Now, by this time, what happens? We see 14 discrete stripes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 discrete stripes, which are like this. You can see. Go from this side, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Segmented body plan is established. We saw the segmented body plan already. Here, embryo looks like this. This is due to, and we know already the genes. For example, gap genes are Kruppel, nerves, etc. Pair rule genes are even-skipped and Fushi-Tarazu. Before even-skipped and Fushi-Tarazu disappear, in this segmentation, this stage correlates or coincides with cellularization. Cells become individual here. There are no more syncytial blastoderm. Here is the beginning of cellular blastoderm. Now, once pair rule genes disappears, they actually activate segment polarity genes, and each cell here, since they become cellularized, I try to draw embryo here. Cells in this stripe, for example, Let's say we are talking about first try. Or we have third stripe. This is second, this is third. Cells, they are very, very clearly specialized. And cells, they are not allowed to mix. There's no physical barrier. This layer of cell is not going to mix up with this one with not this one, or either of these cells. And what we call that, this is the time when segment, this is a segment, no? This line is one segment. We call that segments are getting polarized. Polarity. These cells will remain throughout in life on this side and contribute to a specific region of tissue or organ as compared to these ones, and they don't mix up with each other. And this is done by segment polarity genes. And you know, cell fates are, and here gene segment polarity genes are, for example, hedgehog, wingless, engrailed, and I forgot when they are cell signaling proteins. Now what they are doing, they have divided cells not only into certain segments but also compartmental boundaries. In the absence of any physical barrier, cells are not mixing up, okay? And differential gene expression is fully established by activating genes called Hox genes. Hox genes are called master regulators of development. At this stage, when segment polarity genes have done their job, a certain class of genes which are named Hox genes or homeotic genes in fly, they get active. This is the embryo, you see. This is 10 days old embryo. This is 3 hours old. This is 10 days old adult, sorry. This is embryo. Now, when segment polarity genes are disappearing at segmentation, what happens on the chromosome 3 of fruit fly? If I draw chromosome 3 like this, here there is a region called homeotic genes, and homeotic genes are in two important clusters: bithorax complex, other one is antennapedia complex. Bithorax complex comprises of genes called UBX, ABDA, ABDB, and an antennapedia comprises of labial, proboscipedia, deformed. You don't need to memorize the names. What is important? These genes are expressed in specific segments. Look, labial is expressed only in segment 1. Proboscipedia to the next, deformed here. Sex chromes reduced here, and so on. And you have UBX, ABDA, ABDB, and they are called master regulators of development or selector genes because they finally lock the identity of individual segments. And you see that these segments, their identity is maintained, is fully remembered like a memory of cells. Look all the way to adult. These were these ones. ABDA expressing cells to these. UBX expressing, and so on. We call this phenomenon epigenetic cell memory. Today we are going to talk about epigenetics. The gene called UBX is present here in these cells as well, these ones, but it's switched off. ABDA is present in these cells as well, but it's switched off. ABDB is present in all the cells in the body, but ABDB is only maintained in the active state here and not in any of the other cells. So this is called epigenetic cell memory, how this differential gene expression pattern, which is established early during development, how differential gene expression pattern, which is established early during development, which gives cell identity, how it is maintained throughout the development till adulthood and till our death. You will be shocked. You will be, if not shocked, you will be amazed to see what we learned in fruit fly, in Drosophila melanogaster, is 100% true in you, in me, in all the humans, in mammals, mouse, rabbits, whatever. The way genes are organized on their chromosomes, for example, we humans have 46 chromosomes. The organization of genes is same, but in humans we have 4 such clusters. But organization and the genes are same. Look, this is a mouse embryo, closer to us than flies. Look, ABDB gene, which is similar genes in different species are called homologs. Homologs. So similar genes, ABDB, homolog of ABDB in mouse is B9, ABDA is B8, UBX is B7. The way they are expressed from head to tail, from anterior to posterior, they are exactly conserved, fully preserved in mouse, and the identity of the cell type which they give is also 100% preserved. If now you have misregulation means this gene, which is called antennapedia, which is expressed only here in the legs, okay, and is silent here, or UBX, which is expressed only here and silent there. If the expression pattern is changed, if they start expressing in different regions where they are supposed to be silent, we call this misregulation or ectopic expression. Let's say if you have antennapedia, which is only on here and off there. If antennapedia becomes on here, what will happen? Or UBX becomes on. Look, this is a normal head, normal wild type fruit fly. This is antennapedia. Antennapedia expression in this region lead to legs on the head, legs. Look at this one. Legs. Or this is another beautiful example. You have the master regulator of gene, which is called ILS. Homolog of ILS in mammals is called PAC6. If you switch on PAC6 or ILS gene, ILS is only expressed in eye specific. I-specific gene. If you express I-specific genes, which is I-less, on the legs, you see two beautiful legs on the eyes. You see beautiful eyes on the wings. This is how we understand development using genetic tools. This is how we play with the different developmental toolkit to understand, okay, how cell fates are determined, what are the genes which give identity to cell fates, and then what are the genes which ensure that one cell fate is determined, it's remembered cell generation after generation, so that such a phenomenon should not happen. Or such, imagine your eyes, they start expressing heart specific genes, or your eyes, they start expressing neuronal gene, and neurons start expressing eye specific gene. What will happen? Straight away cancer, death, abnormality. These experiments show you this, how crucial this is to maintain that differential gene expression established early during development. The Hox genes. We already talked about what the Hox genes are. At molecular level, the Hox genes are basically they contain a specific region in their proteins which is encoded by 180 base pair DNA sequences, and in protein we call this region homeodomain. And this homeodomain or homeobox region is this 180 base pair region. So at the gene level, let's say at DNA level, you have this 180 base pair region, messenger RNA is made, and then this protein is made where you have, let's say this is the region which is the homeodomain. Now this homeodomain, and these Hox genes are basically transcription factors. They go bind to DNA and switch on or switch off a gene, and they're conserved throughout the evolution from flies to humans, even in plants. And if you look at plants, when I say plants, you must have been surprised, okay, in flies they give rise to eyes and in legs or whatever, and humans also have. So what they are doing in plants, what is their role? So it's beautiful. So the Hox genes basically are involved in pattern formation and symmetry, and they discovered 3 classes. So like they are different than Bithorax and Antennapedia, but these are the Hox genes in plants are categorized into 3 categories or 3 classes: gene of A class, gene of B class, and C class. They meet each other as protein, bind to DNA, activate some genes. They may inhibit each other. They may facilitate each other. So this was another beautiful genetic screen in plants, which has left a huge impact in the field of science. And that is that, you have what they discovered, that genes A, class B, and class C, they have a role in flower development. And we know that flowers have 4 whorls. If you carefully, closely look at flower, so flowers have sepals, then petals, then stamens, then carpels. This is sepal, the very tiny leaf the green color leaf, if you look at rose, आज कोई भी फूल पकड़ के देख लें, रोज देख लें, उसके बाहर वाली साइड पे सबसे छोटा सा एक ग्रीन 5 लीव्स होते हैं, ठीक है. This is sepal. Then comes the big red colored, the petals. Second whorl. Okay. Then the inner whorl is the stamens, and finally you have the carpel. And they are organized like this: sepals, petals, stamens, carpel, carpel, stamen, petals, sepal. If you go from one side, you come from this side, okay? Go from this side, sepals, petals, stamens, carpels, carpel, stamen, petals, sepals, okay? Whorl 1, whorl 2, whorl 3, whorl 4. Elliot Myrowitz's lab did a beautiful genetic screen in Arabidopsis to understand how this symmetry, this pattern is formed. And he discovered 3 different classes of genes, 3 different classes of phenotypes. As a result of genetic screen, you always first find phenotypes. And those phenotypes were similar, okay? And he categorized those phenotypes into 3 different classes of mutants. For example, he said the normal Arabidopsis flower looks like this. This is the sepal, outermost whorl, sepal, stamen, and carpel. Sepals, petals, stamens, carpels, carpels. This is a symmetry in flower. He said a class of mutants, many mutants, they have found which have lost actually the sepals altogether, as well as petals. They only have stamens and carpels, no sepals and no petals. Many mutants, he grouped them together, and he said these are class A mutants, just based on phenotypes. The next phenotype they observed was sepals are there, petals are missing, stamens are missing, and carpel is there. There are 2 carpels, look, carpels and carpels. And similarly, sepals also, sepals and sepal, 2 whorls of sepals. Instead of 1 whorl of sepal, they have sepal, then carpel, then carpel. So stamen and petals are. And finally, they name them, okay, class B mutants or gene B mutant. And the last one they found, you know, the stamens and carpels are altogether gone, and you have multiple whorls of petals, and below them you have, so 1 sepal and then petals. They name them class C genes. Now they made a model, and they said, okay, in normal wild type, if we have now phenotypes of A, class B, and C, they said the gene A is actually in normal flower, normal wild type. Gene A is expressed in whorl 1 and whorl 2. I tried, yeah, here there is whorl 1. This is whorl 1, whorl 2, whorl 3, whorl 4. Sepals, petals, stamens, carpel. They said the gene A expresses in sepal 1, in whorl 1 and whorl 2, and the class B is expressed, gene B is expressed in whorl 2 and 3, and C is expressed in 3 and 4. How they could conclude this? They concluded this, look at these results. From here, they extrapolated the expression domains of A, B, C. Look, when you have A is gone, gene A is gone, what they found, the sepals and petals, they are replaced by carpel. Petal is also replaced by carpel. It means A is something to do with C. So what they said, A is actually inhibiting spread of domain of C. A blocks, but when A is gone, C spreads all over. And when you have A alone here, you will get sepals, but whenever you will have A and B together, you will get petals. And whenever you will get B and C together, you will get stamens, and C alone gives you carpel. Now, in the absence of A, when C spread all over the four worlds, what do you have? C alone instead of A gives you carpel. C plus B gives you stamens. B plus C again stamens, and then carpel. And you go from here, carpels, carpel, stamen, stamen, carpel, carpel, stamen, stamen, carpel. And you can conclude this for class B gene, class C gene. Okay. When you don't have B, there's nothing. A plus B together give you petals. Now you have only A, you will get duplication of sepals. Sepal, sepal, and 2 times carpel. Because B is absent, together with B and C, they are supposed to give you stamen. And when C is gone, now A is going to spread into the domain of C, and A alone in its normal domain will give you sepal. Wherever it will be together with B, it will give you petals. So instead of stamen, due to absence of C, so stamen only comes when B and C are together. Now you have B and A, you will have petals again. So duplication of petals and then duplication of sepals. So this is called the ABC model of flowering. We know now it was discovered in early 90s, mid 90s. And now at the protein level, it's characterized as well.
Sir, I have a question.
Yeah.
So in this model, the mutants that we saw, do they lead to like plant death or are they just variations in species?
No, they die. It's very difficult to maintain them.
Okay, okay.
Even these ones are very difficult to maintain. So there are special tricks we use to maintain in our labs. So my lab at LUMS, we actually, if you haven't seen my lab at LUMS, may Allah, Corona goes down quickly and we are back to campus soon. So you will see we have this fruit fly lab and we are doing a lot of work which involves actually development as well as the Hox genes and, you know, my PhD, one of the PhD is defending thesis tomorrow. And, you know, we have characterized these things. So these are difficult to maintain these mutants. So now let's come, our topic was differential gene expression linked to cell identity. Now we are arriving at the stage where we are linking everything we have learned today with the very first lecture, the central dogma, the differential gene expression, and I promised you we'll talk about epigenetics. There's a subject very close to my heart because this is my expertise as well, and this is my passion. We work on epigenetics, cell memory, cell identity, cell fate determination in fruit flies at LUMS. So we said differential gene expression. Remember, differential gene expression is maintained early on development. And you know, the cells, they remember their identity, all these cells. Yeah? Is that a question? So we said that these cells mitotically keep dividing all the way to adulthood. And you know, you end up maintaining the cell fates and cell identity. Now, how all this at the level of transcription happens? I'm not going to go into the details of transcription because we already learned about transcription, but we'll quickly have a recap. But we'll spend more time on epigenetics today. So transcription in eukaryotes. So DNA is wrapped around histone proteins. You know, there are 8 histone proteins. We call this histone octamer. And there are 2 molecules of each. So there are 4 different kinds of histones. H2A, H2B, H3, H4. And 2 molecules of each, they make this histone octamer. 146 base pair of DNA, double helical, goes like this in 2 rounds. Then the next such structure. Then the next. One single 146 base pair plus histone octamer is called a nucleosome. And that's how your chromosomes are organized, packaged. This is shown here. This is one nucleosome, 146 base pair plus this. Now, in eukaryotes, in humans, in yeast, you know, we have multicellular organism. We are multicellular bodies. A human body has more than 200 different cell types, for example. And it's difficult for a single RNA polymerase to, you know, perform transcription or all the jobs within the cell. So what eukaryotes have done, you have 3 different kinds of polymerases. RNA polymerase 1, which transcribes all the ribosomal RNA. You remember ribosomes? They had, I told you they have RNA as part of their physical structure. RNA polymerase 2 is the one which encodes all the genes which are transcribed into messenger RNA and then translated. So RNA pol 2 performs this function. And RNA pol 3 transcribes all the tRNAs. You remember? Clover leaf shape, small RNAs which contain anticodons and corresponding amino acids. Now, for this particular lecture, we'll mainly focus on RNA pol 2 mediated transcription. So transcription in eukaryotes, if we go in detail, you know, you have Tata box, the promoter region. So gene is basically promoter, and then the gene body. Gene body is what? Which contains in eukaryotes exon, intron, exon, etc. And also the transcription initiation site. So RNA polymerase 2 cannot bind by itself. There are many transcription factors, and one of the most important one is, you know, there's a protein called TBP, Tata T8TA binding protein, which recognize this Tata box, and it brings in another protein which is called TF2D, transcription factor 2D, and then a stepwise organization of, you know, RNA polymerase 2, then they recruit together. Now RNA polymerase 2 will come, a big complex of RNA pol 2 is assembled onto promoter element, and this is called transcription initiation complex. When everything is bound on promoter, we call this transcription initiation complex. Sorry. The RNA polymerase 2 undergoes some phosphorylations, and then eventually RNA pol 2, it starts sliding through the gene body, and they start making messenger RNA. Okay? When RNA pol 2 starts moving through the gene body, we call this transcription elongation. Messenger RNA is elongating, and then when it slides off, we call this transcription finally termination. This is a normal general process of transcription. However, when we say genes are selectively switched on and switched off, you know, in response to these morphogen gradients and I don't know, different transcription factors, the eukaryotic genome is very interesting. It's not just promoter and coding sequence gene body. It contains sometimes far away from the gene. For example, this is the gene body of a gene. This is the promoter element. Far away, these broken lines represent far away from the gene body. There are DNA sequences which are called enhancers. These are, remember, enhancers are to DNA sequences we write elements and to proteins we write factors. So enhancer elements are DNA elements which attract certain proteins which when bind, they have a positive effect on RNA pol II complex, and this positive impact then lead to transcription or synthesis of messenger RNA. In the absence of enhancer bound by activator proteins, you won't be able to have transcription of cell type specific genes. So these enhancers, they are actually very crucial in cell type specific gene expression, differential gene expression. Now you will be surprised that, and you should know this, you should think about this. I told you UBX gene is present. For example, this gene, UBX, is present in every cell in the body. From here to eyes, everywhere is in the nuclei, UBX is present, but UBX is only expressed in this particular region, the green one. There are these enhancer elements for UBX, which the proteins for which bind to these enhancers are only present here and absent in these regions. Or in these regions, the repressors, they dominate and they block the expression of UBX here. So we call this, look, this is a long distance interaction. DNA bend over backwards like this because physically it's far away. So now the activator comes closer to RNA pol to provide it necessary push and transcription initiates. How this
sir,
yeah,
sir, slide 6 हो गई है. So we are seeing the Hox genes वाली slide.
क्या हो गई है?
सर मैं Hox genes वाली slide आ रही है if you are at the transcription.
अच्छा, let me. Now you can see this. Now you see activators and repressor slide. Which slide do you see now?
Sir, it's not. Yes, sir. Now it's.
Yes, sir, we do.
You still don't see activators and repressors?
Sir, we do
Sir,
see
अब
it.
आ रही है.
अब आ रही है.
ठीक है. So in order to bring this here, you know, there are very specialized proteins which are called chromatin remodeling or DNA bending proteins. And just like you have enhancers, there could be regulatory elements where, you know, another protein which is a repressor, which instead of activating, instead of having a positive effect, it may have a negative effect, and we will call that repressor, which represses or blocks the transcription of RNA pol 2. So this is the architecture of chromatin in which we are going to learn epigenetics, and in which we are going to learn, you know, how differential gene expression patterns are established or maintained, etc. Now, when in addition to these regulatory elements which are called enhancers, etc., there are other things as well. Oh yeah, I forgot one thing here. Here, in addition to 4 histones, when DNA between, so this linker DNA, we call this linker DNA. Linker, the DNA which is not wrapped around histones. This is bound by a 5th histone, which is called H1, histone H1. Okay. Now, in addition to, let's draw it here. Let's say we have now nucleosomal conformation of DNA. DNA goes like this. If I zip apart DNA and histones, so what we have, this is how DNA component of chromatin, all this is chromatin, no? This is the DNA part of chromatin. And then we have the histone octamers. We just learned about enhancers. Let's say here is an enhancer element of a gene. Far away there was this promoter element, and then you have the transcription start site. We just learned, you know, enhancers, they are DNA sequences, and activators are proteins, and activators will have some very specific amino acids which recognize these nucleotides or DNA sequence in the enhancers, or same as vice versa for repressors. What we discovered in the middle of, you know, 70s or so, that DNA sequence, let's say you have A, T, G, C, A, T, G, A, C, G, G, C, A, T, 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 A, C, G, C, G, T, A, T, A, T, A, T, A, the Tata box, okay, and so on, the sequence. What we discovered that the cytosine, in a very specific context, which is CG, if DNA is going from 5 to 3 context, this will be symmetrical sequence, no? CG and CG, mirror image sequence. You know, the cytosine, it can be methylated. A methyl group is attached at the cytosine. Look this. Cytosine is what? A base. You have deoxyribose. I draw it here. The cytosine base, okay, the deoxyribose, and the phosphate group. This is a nucleotide. So DNA sequence in such context, which I drew on the last slide, and it's here as well. In symmetrical sequence, it can get a methyl group attached to the 5th position of in the nitrogenous base, and we call this, such a cytosine is called 5-methyl cytosine. We also write it like this. This was an interesting discovery, that cytosine can be covalently modified, covalent modification of cytosine. It was not enough. They discovered that if you have a gene let's say this is the promoter region of the gene. If cytosines in CG context, they get methylated, this gene is going to be silent. Hard sign mean messenger RNA cannot be synthesized. RNA polymerase cannot bind. Even if it is bound, it cannot transcribe. Gene is silent. We call this gene is off. In contrast to off, if you have to now switch it on, you have to get rid of the DNA methylation, and only then transcription will take place and gene will be on. This gene we will call active. Gene is active. This one we call gene is silent. Here you are off, here you are on. So they discovered that if the promoter region gets methylated in the cytosine sequences, the gene is going to be switched off. In molecular biology, cytosine is also, it's referred to as 5th base. Molecular biologists, they call this as a 5th base, although it doesn't make sense because there are only 4 bases: adenine, thymine, guanine, and cytosine. But since methylation has very, very specific consequences on the activity of the gene, you can turn off a gene. They call this, this is the 5th base. Although this is not a base. Now, they also discovered that, there's a specific DNA methylation machinery. There's an enzyme which catalyzes, which methylates cytosine. It doesn't come by itself. There's a protein called DNA methyl transferase, which adds methyl group here covalently, and once it's added in CG context, here for example, when this DNA will replicate, look, this is now blue, these are the mother strands, for example, the mother strand and the mother strand, but the daughter strand is not methylated. The symmetrical sequence, we call this state hemi. Hemi means half. Hemi methylated. Okay. So this hemi methylated state is recognized by another enzyme which is called maintenance methyl transferase. Maintenance of methylation. This enzyme is different than the first one. The first one is called de novo methyl. It's also an enzyme transferase. De novo methyl transferase, what it does, it adds methyl group on unmethylated DNA where there's no methylation. Maintenance one, it recognizes hemimethylated DNA, and then it adds methylation on the newly replicated DNA strand, and you convert hemimethylated DNA to fully methylated DNA. And eventually what happens, you have both the daughter strands contain fully methylated gene, and gene will remain off. Okay, gene will remain off. So if in mother cell this gene was off, in both the daughter cells gene will inherit as in the off state. We call DNA methylation as the epigenetic modification, or we call all this epigenetics. Because why we call this epigenetics? Because epi means above. Okay, nothing is happening, so the sequence of the gene remains same. Imagine this is double-stranded DNA I drew. Nothing happened at the DNA level. The sequence is just like wild type sequence. No change in the sequence of the DNA. It is just like original sequence in the original DNA. No change in the DNA sequence. But epi means above. Above the level of basic DNA sequence, some modifications have happened, which have resulted in silencing. Silencing means gene function is lost for the time being as long as DNA methylation is there. So we call this epigenetics, that you can change gene expression without causing any mutation or any change in the DNA sequence. It can be DNA methylation. It can be also modifications of these histones. Histone proteins, we are soon going to see their modifications. So now, if we go back to our differential gene expression during development, now imagine you started your life as zygote. You went to different cells. Cell fates are determined. All the genes from A to Z of this, all the nucleus contain, all the cells contain same set of genes, but they are differentially switched off and switched on. Now, let's say you have here, we just take to gene A. A is off here, on here, on here, and on here, and off here. Now, this differential gene expression will only be possible in DNA methylation. All this has happened at the level of epigenetics. So promoter region of A will be methylated here, will be not methylated here, not here, not here, and methylated here. Here you will have gene A in the silent state, and here on. And when this cell will divide into two cells, maintenance methyl transferase will ensure A remains off. All the other genes which we are for the time being ignoring. Here, because there's no methylation, there's no role of maintenance methylation, so daughter strands will have on. Here again, since it's methylated, the daughter cells will have it in the off state because DNA is methylated. So this is epigenetics. You play with, the epigenetic modifications without changing the primary nucleotide sequence of gene. The function of gene is kind of lost. It's silent. No messenger RNA is being made. Protein is not there. It's like, isn't it like a mutant-like state? So that's why they call it epigenetics. However, an important criterion for epigenetics is this thing, that once you have switched off a gene, it goes through cell cycle, through mitosis, and is inherited to future generation. We call this epigenetic inheritance. Why we call this epigenetic inheritance? Because together with the 46 chromosomes in these cells, the gene expression state due to DNA methylation is also being inherited from mother to daughter cell. So that's what we call epigenetic inheritance, which was here. So differential gene expression when cell fate determination takes place early during development in mammals, in humans, or in plants, genes are differentially switched on and switched off due to these epigenetic phenomenon, and eventually what you see, you have epigenetic inheritance and genes are differentially expressed. In addition to DNA, the histone proteins, they also get differentially, they also get covalent modifications. But like in case of DNA, we had only DNA methylation. In case of histones, this is just, we focus on, for example, just histone H3. This is the N-terminal tail of histone H3, okay? Now, and this is one nucleosome. We are showing one nucleosome, okay? Look different amount. This is the N-terminal side of protein, and this is the carboxyl terminus. Proteins are made up of just amino acids. Okay? So you have, for example, arginine 2, lysine 4. There are intervening amino acids as well, but we are only seeing amino acids which are modified. Lysine 9, serine 10. These are different amino acids in a protein. I just for simplicity show you lysine. Just focus on lysine 4, lysine 27, lysine 9. All the lysines in 4, 9, 27, 36, they get methylation. We call this, let's say this one is called H3, histone H3, lysine 4. 3 methyl groups can be added. This is a lysine in the histone, okay? And lysine can also be acetylated. So A means acetylation. And this is acetyl group is added onto lysine amino acid. If you have lysine 14, lysine 16, whatever lysines are acetylated, they correlate with on state of genes. Genes are on. They are responsible for genes to be maintained in the on state. Lysine methylation is context dependent. For example, lysine, histone H3, lysine 4 methylation, gene will be on. If you have histone H3, lysine 9 methylation, they are counteract, exclusive. They counteract each other. Now gene will be off. Just like DNA methylation. And histone H3, lysine 9 methylation and DNA methylation, 5-methyl cytosine, they go hand in hand together. They are together to maintain genes in the off state, silent. Now, lysine 27 methylation is very beautiful phenomenon. Why? Because lysine 27 methylation is involved in silencing of developmental gene. The Hox genes, you remember? Homeotic genes. These genes, they are silencing involves lysine 27. Wherever they need to be switched off, they will be lysine 27 methylated. And wherever they are to be switched on, wherever they need to be switched on, lysine 27 methylation needs to go and will be replaced by acetylation. There is very important point. So we are now coming down to the last phenomenon of differential gene expression during development. So these are key developmental genes, and let's stick to just Hox genes as our best friends here. Humans, so flies, एक important point है, flies do not have DNA methylation in their most of life. They have DNA methylation only for first 30 minutes or so during their life, but they do have histone methylation, everything histone regulated. So we call them, this phenomenon is called epigenetic gene regulation. It involves DNA methylation, histone modifications. When you have, for example, lysine 27 methylation or lysine 9 methylation or lysine 4 methylation goes for active genes, all the active genes, and they are exclusive. They inhibit K27 methylation, and K27 inhibits lysine 4 methylation. So when you have either the DNA methylation, when methylated cytosine is there, this is recognized in DNA sequence. This is recognized by other proteins which recognize cytosines or DNA when it is methylated. And these proteins, they are the ones which convert methylated cytosine into a silent state of gene. Same is the case for here. If you have lysine 4 methylated, histone H3 lysine 4 methylated, this will be recognized by specialized protein complexes which are called trithorax group proteins, and they maintain genes in the on state. And if you have lysine 27 methylation, this methyl mark at H327 is recognized by protein complexes we call PCG, polycomb group. Polycomb group. They are the ones, polycomb group proteins are the ones which maintain Hox genes, UBX in the silent state. Polycomb group is going to maintain genes in the silent state wherever they are supposed to be silent. And the trithorax group genes, which are these ones, trithorax proteins, they recognize this H3 lysine 24. They maintain the Hox genes in the on state wherever they need to be maintained. And that's how differential gene expression pattern, you know, I take you back here. Here, at this stage, polycomb group and trithorax group take over the job. Polycomb group maintains Hox genes in the silent state wherever in the body they are supposed to be switched off, and trithorax maintain them in the on state wherever the on state of these genes is to be maintained. And that's how cell identity is maintained. That's how cell fates established early during development is maintained. One phenomenon which you will be happy to learn is, and which is linked during epigenetics, is X chromosome. So we learned in genetics that there are 2 X chromosomes in females, and in males we have only 1 X chromosome and the other Y. So it means between females and males, there is difference. Is there a difference between males and females? Yes or no?
Yes, sir.
Females, may I get an answer from you?
Yes, sir.
So there is difference in the gene dosage. You have 2 X chromosomes here. All the genes which are on this X, they are here on X as well. In male, only you will have dosage of genes 50%. In female, you have 100%. On the Y, you don't have X chromosomes. So discussion दूसरी तरफ चली जाएगी. Males and females are not equal. समझ आई मेरी बात? Females are more superior than males. They have more gene dosage. So what females do, they say, बेचारा मेल क्या करेगा, सारी जिंदगी कॉम्प्लेक्स में रहेगा, चलो let's epigenetically reduce the gene dosage, and they inactivate one complete X chromosome. उनके 2 X chromosome होते हैं इस तरह. This is one X, the second X. What they do, they say, okay, we keep all the genes on one, and we jumble up the second X chromosome, like literally jumble up. It looks
So
like
when
this.
does this happen?
Can you see this? This dot? This is called bar body. It was discovered by scientists whose name was, I don't completely remember, something bar. So all the genes on one X will be now silent, and this is done by polycontra-thorax using H3K27 methylation, DNA methylation, etc.
Sir, at what time, like in life, does this happen?
Look, this is what is happening. Methylation and whatever, and gene dosage become equal. They have to do this. There's no choice. If they don't do this, they will die, because human machinery is programmed to tolerate only 50% of the gene dosage of X chromosome. It happens very, very early during development. Like, Abdullah, can you Google quickly? I think it's embryonic stage 5, or let me admit my lack of knowledge at the moment. I can't recall. I think it's embryonic stage 5 or 8. Abdullah, can you Google quickly and then let us know? Very early development, when you are still few cell stage. And it's also random. Random means what? Let's say you are a couple of cells, females. All cells contain 2 X chromosomes. One of the X came from father. Is it true? And the other X came from mother. Is it true?
Yes.
What it does, it is the very, very precise developmental stage. It's either father or mothers in different cells. It doesn't matter which one. It randomly switches off one. In some cells, maybe father's copy goes off, in others, mother's copy goes off. And you should go and read. It's such a beautiful phenomenon. Okay. And you have eventually gene dosage, and all this is, it involves 5-methyl cytosine. It involves DNMT1. You remember DNMT1 was maintenance methyl transferase, the enzyme which generation after generation maintains CG methylation in DNA. And it involves polycomb group proteins, which are involved in H3K27 trimethylation. Isn't it interesting?
Yes, sir.
It is interesting.
So does this mean that, sir, before birth, like every human has a higher probability of being a female rather than a male?
Very good that you asked this question. I wanted to talk about this when we talked about X and Y, and I simply missed that. So remember, after meiosis, the 4 gametes females produce, they are all equal. X क्रोमोसोम होंगे ना फीमेल में, 22 तो होंगे ऑटोसोम्स, मैं सिर्फ यहाँ पे X क्रोमोसोम बता रहा हूं ना, ठीक है। 22 होंगे ऑटोसोम्स and 1 X chromosome होगा. What will be the male after meiosis?
2x 2y.
When, and its random process of fertilization is random. Female is not responsible for, she is providing uniformity. All her oocytes, and she makes, in one menstrual cycle, you have just one oocyte, and it will always be X. The male sperms, they will be either X or Y. It depends which one of the sperm finally hits. So if this one hits, we will have 2X, a daughter. If this one hits, we will have XY, which is a boy. So who is responsible for sex determination of the baby?
The male.
It's the male. In our society, and I get really upset. I get really upset. It's disheartening. Not only in illiterate societies, regions of Pakistan, unfortunately, in educated, so-called educated, I don't call them educated, so-called educated people, if the baby is a boy, is a girl, and in a family repeatedly they have 2 or 3 girls, I don't want to recall those incidences, what I have seen in my life. You know, they don't congratulate. आजकल तो ये हो गया है, अल्ट्रासाउंड हो जाती है, वो 6th ट्राइमेस्टर में पता चल जाता है. उर्दू में कहते हैं उनकी मां मर जाती है. And after some time कुछ तो उस एक्सट्रीम पे चले जाती हैं कि जी I don't want to go in this debate. I wanted to talk about this. It's very important for you. As a teacher, I think my job is not just talk about textbooks. My job is to talk to you about, you know, the context in which your knowledge, your education should give you, which should help you, you know, look at things in much bigger picture. And you know, they start thinking about कि जी नस्ल कैसे चलेगी और कि पता नहीं जैसे ना बेटे ना हुए तो क्या होगा. And this is male. अव्वल तो जो simply it's controlled by nature. It's not even fault of yeah male. It's not fault. मैं तो ये जो feminism की debates है ना, और जो ये जो male और female की equality है, मैं तो उसके वैसे ही खिलाफ हूँ. Why? There are many things which, for example, I have no control on my birth. Did I decide to, you know, be born in an educated family? Did I have any control on my birth कि जी मैं एक अमीर के घर में पैदा हूँ या गरीब के घर में पैदा हूँ? Did I have the chance to be a boy or a girl?
No, sir.
My parents had a chance to decide. कुछ भी नहीं है.
No, sir.
ठीक है. और इंडिया में तो उन्होंने अल्ट्रासाउंड बैन कर दी है. वो अल्लाह माफ करे, वो जो हम 1400 साल पहले बुक्स में पढ़ा करते थे, जमाने जहालियत की चीज के बेटियों को जिंदा दफन कर देते थे, वो काम शुरू हो गया था. ये रिसेंटली 2, 3, 4 साल पहले आई वाज फॉलोइंग. तो ये बेसिकली क्वेश्चन ये था कि डू वी हैव इक्वल प्रोबेबिलिटी टू बी अ बॉय और गर्ल? यस, वी हैव इक्वल प्रोबेबिलिटी. इट डिपेंड्स के कौन सा वाला हिट करेगा, ठीक है।
सर स्पर्म सेल्स के अंदर दोनों एक्स और वाई क्रोमोसोम्स दोनों होते हैं, वो बस ये डिपेंड करता है कि हिट कौन सा करेगा।
हाँ नहीं, सारे स्पर्म्स में इक्वल होंगे ना, या एक्स होगा या वाई होगा,
या वाई होगा।
ठीक है। एक ही स्पर्म में एक्स और वाई इकट्ठा तो नहीं ना होगा, क्योंकि ये तो मायोटिक सेल है ना, हैप्लॉइड है, ठीक है।
सर
तो
सर
उस
हैप्लॉइड इज 4 हाफ सेल है ये।
हैप्लॉइड या दिस वाज अ डिप्लॉइड,
यस सर, दिस इज
2n,
अ डिप्लॉइड बस, ठीक है सर, ठीक है।
46 क्रोमोसोम्स, मायोसिस हुई, मायोसिस 1 में 23 होमोलॉग्स एक साइड पे चले गए, 23 एक साइड पे चले गए।
जी
मायोसिस 2 हुई तो ईच सेल गेव यू 2 सेल्स, तो वो ये वाले आ गए,
हाँ जी,
ठीक है। तो ये इन्होंने जाके फर्टिलाइज करना है।
ठीक है सर।
सो डोंट टेक मी रॉन्ग व्हेन आई से कि आई एम, मैं आपको एक और एग्जांपल देने लगा था, ये बड़ी प्यारी है, ये भी पेरेंट्स से ऑफस्प्रिंग पे, ये आखिरी स्लाइड है, कहीं टाइम तो नहीं ऊपर हो गया? ओह
सर टाइम 15 मिनट ऊपर हो गया, इनफैक्ट
I didn't
20
see it.
minutes.
Okay, we stop it here. So there are things which are decided, programmed by nature, and we don't have any control. And this whole debate के मेल्स और फीमेल्स जो हैं वो इक्वल हैं, और जनाब वो नौकरी क्यों करे और वो नौकरी क्यों ना करे, और these are purely decisions between husband and wives. Me and my wife both are scientists. She was a neuroscientist. I was a epigeneticist, और जो मर्जी मुझे आप नाम दे दें. And you know, we both did. We lived in Switzerland. So, but both boys and girls, they have, the parents have equal responsibility and duty to educate them both equally. Both should be given equal opportunities. And you know, later on, if husband wife says कि यार दोनों ने जॉब करनी है, दोनों करें, अगर किसी ने कहना, आई नो माई फ्रेंड्स जिनकी वाइफ जॉब कर रही हैं, उन्हें कहा, और हस्बैंड ने कहा यार यू आर बेटर, यू आर डेंटिस्ट, यू विल अर्न मच मोर एंड आई टेक केयर ऑफ किड्स एट होम, या दोनों ने कहा नहीं हम दोनों मैनेज कर लेंगे, तो ये पूरी जो एक डिसीजंस होते हैं, दे आर बेस्ड ऑन अ गुड friendly relationship between husband and wife, and respect and opportunities and rights पे कोई compromise नहीं होता, whether you are a boy or a girl, respect पे कोई वो नहीं है, respect आपकी equal है। और बल्कि वो जब ये equality की बात करते हैं तो मैं पहले तो कहता हूँ इन लोगों को कि ये unequal है, क्योंकि biologically भी unequal है, दोनों की biology भी different है, but eventually I tell them कि look, female is like here and male is like here. Female is much bigger, रुतबे के हिसाब से, डिग्निटी के हिसाब से, वो माँ होती है, ठीक है, वो जनरेशन क्रिएट करती है पूरी की पूरी, उसको वो एक पूरी नेशन क्या पूरी जनरेशन बना देती है, और बाप का रोल कम नहीं होता, इट्स इक्वल। बट माएँ जो होती हैं वो नबियों को भी उन्होंने जन्म दिया, पेट में रख के जन्म, जो माँ का रुतबा है वो खुदा के करीब है, खुदा ने अपने आप को माँ से कंपेयर किया कि मैं तुम्हें 70 माओं से ज्यादा प्यार करता हूँ, बाप गरीब को तो लिफ्ट ही नहीं करवाई, तो माँ का रुतबा तो बहुत ही, या फीमेल्स का, गर्ल्स का रुतबा तो बहुत बड़ा है, उसको अगर हम कहें कि नहीं वो मेल्स के बराबर है, तो मेरी डिक्शनरी में हम उसे लोअर डाउन कर रहे हैं। अदरवाइज तो जो राइट्स एंड इक्वालिटी, ये वो जो चीजें होती हैं उस पर तो कोई है ही नहीं है। सरकार दो आलम सल्लल्लाहु अलैहि वसल्लम ने, प्रॉफिट मोहम्मद सल्लल्लाहु अलैहि वसल्लम, ही गेव अस वेरी क्लियर इंडिकेशंस के, और याद रखना बेटियों को एक तो जिंदा दफनाते थे ना वो कि जी गर्ल्स हैं, बेटी की शादी कर देना उसकी मर्जी के खिलाफ, ये भी उसे जिंदा दफनाने के मुतरादिफ होता है, अगेंस्ट हर विल या अगेंस्ट हर विलिंगनेस, वो जो एक कि नहीं जी नहीं यहीं पे कर दें, जिस मर्जी रीजन की वजह से आप कर दें, इट्स इक्विवेलेंट टू बरिंग हर अलाइव, क्योंकि वो जिंदगी एक मुर्दा की गुजारती है उसका तो, और बेटी की जो शादी होती है वो तो उसकी कंसेंट से होती है। ये मेरी बातें याद रखिएगा।